Welcome you all to this Cryo-EM course. Last class we are discussing about the thermionic gun and field emission gun. So, we will continue our discussion, and in this particular case we already discussed about the differences between the thermionic gun as well as the field emission gun. So, let us take a look little bit what is the difference. So, last class I showed you how the electron extraction is happening, what is the crossover point, what is D0. So, this is a diagram taken from the book where you can see this is the filament which is connected with the electric current, and these are the Winot cylinder, and this is the crossover point. And the field emission gun, this is the first anode and second anode, and then heat energy is using and we are getting the electron. So, this is the two major difference. I put them side by side so we can see the differences between them. In field emission gun, what is the field emission gun? So, field emission gun is much simpler than thermionic gun. In order to get a field emission gun to work, we make it with the cathode with respect to two anodes. The first anode is positively charged by several kilovolt with respect to the tip, and this charge produce the extraction of the voltage, since it generate the intense electric field which enables electrons to tunnel out of the tip. Increasing the extraction voltage when we first switch on the gun has to be done slowly, so that thermomechanical shock does not fracture the tip, because most of these tip are made by tungsten filament and then some kind of coating. So, those filaments are very expensive. If we just consider, last class also I discussed about the tungsten filament, Schottky filament. Schottky filament I told it is mainly called the XPEG. Then we are talking about the field emission gun. So, if we just compare the why we are worried about the lifetime of any filament. So, filament's lifetimes is very important, because tungsten filament is comparatively cheaper. So, if you just consider near around 18 to 20,000 rupees you can get one tungsten filament. But as we use very strong heat energy, and that is going to damage the tungsten filament very quickly, and you have to replace the filament very frequently. So, replace the filament means you have to change the gun filament. So, if you remember at beginning when I was talking about the entire column, it will stay under high vacuum, and if something is staying under a high vacuum, even your gun will also stay at high vacuum. You have to maintain that high vacuum. So, even if you have to change the filament, you have to break the vacuum of the gun separately, you have to block the column separately. If this is your column, you have to block it and open the gun and then change the filament, and then you have to go and fix the upper part of the microscope again, and then you have to get the actual vacuum. If you do not get the actual vacuum, it cannot generate the proper electron beam. Your brightness it will be very low and your filament will be damaged. So, you can see it is a time consuming, and very frequently you have to change the filament. And also for the tungsten filament, that beam will be not that coherent. That is one of the major drawback. Price is less, it is cheaper, but every 2 weeks, 3 weeks, 4 weeks, sometime within 2 months you have to change the filament. Now, it is coming the another thermionic emission. If we just consider, that is the lab 6 filament. So, lab 6 filament, it is called lanthanum hexaboride. It is a crystal, and its work function is much lower than any other filament. So, we will discuss about that lab 6 filament and the XPEG also. But when you have a XPEG, if you just consider the price wise, definitely XPEG is much higher price than your tungsten filament. It is almost 7 to 8 times higher price than normal tungsten filament, and the cold FEG is almost 5 to 10 times higher price than normal expect. So that is the reason it is better to operate all this filament at a very high vacuum and maintain it properly. Now one question it is coming that what kind of filament do I use when we'll do the imaging. So here there are some values are there. So for the tungsten filament you can see the work function is 4.5, at the lab 6 filament it is 2.4 and Schottky it is 3 and the cold fake it is 4.5 again. So how we are making a Schottky filament? Schottky filament generally it is a normal tungsten filament. So this normal tungsten filament is coated with zirconium oxide. So if we just consider, so this normal tungsten metal, they make a group where that zirconium hydride powder will be there, and then we are using a very high heat energy, so something 1800 Kelvin, and it will giving a chance to oxidized, which will coat the upper part of the outer surface of the filament, and it will be zirconium oxide. When it will coat with the zirconium oxide, in this particular case work function is drastically reduced, and this lab 6 filament definitely when the work function is drastically reduced, it will really help to extract the electron from the filament tip and get a much more coherent electron beam. So if we pay attention to this, definitely lab 6 filament is suitable and it is better than tungsten filament, but there are much more advanced filament. It will be Schottky filament, because this beam coherence is superior for Schottky fag or cold fag. So Schottky fag, if you just look at the Schottky fag, there is a, as I mentioned, the tungsten filament will coat it with zirconium oxide and it will reduce the work function and it will help to extract the electron properly, and the brightness, if you look at the brightness is much better than the lab 6 filament, and that is one of the reason. And as well as the energy spread, if we look at the energy spread is better, and that is the reason we focus on this either Schottky filament or cold fag for our cryo imaging. Now the question it is coming, in what particular case do we use the lab 6 filament or Schottky filament or the cold fag? Majorly for any kind of room temperature imaging, that is if we just consider that room temperature imaging means any DNA, protein, RNA or any cell that we are using the cryo normal ultramicrotome, we did the section and then we want to image those sample. So better to use the lab 6 filament. There is no need for field emission gun. Field emission gun is very important when we will do cryo-electron microscopy. Why it is so important? Because in cryo-electron microscopy we are not using any kind of staining solution. So sample it will be only in your buffer, and there is a difference between contrast difference, it will come from the difference between the density of your biological science sample and your buffer sample, and that is the reason we need much more brighter brightness, as well as we need a very coherent electron beam, because we are talking about to characterize those biological science sample at a atomic resolution. So we need a very coherent electron beam. So that is the reason for cryo-electron microscopy we mainly use the Schottky filament or cold fag. For normal room temperature temp we mostly use the lab 6 filament. That lab 6 filament is giving a good contrast, we can use any kind of electron dose. One more thing we have to remember, for the cryo sample we cannot use a very strong electron beam, our electron dose will be very limited. So that is the reason contrast will be always poor for the cryo sample, and that is the reason if we use the field emission gun it will be always helpful. For cryo it is very difficult to try with tungsten filament or lab 6 filament, but for room temperature temp mostly we are using heavy metal staining solution. So that is either uranyl formate or uranyl acetate or some other heavy metal salt. As well as we can use a very strong electron dose also. Most of the time at room temperature temp those biological science samples are already dried, so it is not going to damage by that electron dose. That is the reason even if we are using 100 electron per angstrom square or 200 electrons per angstrom square, it is not going to damage that biological science sample. So that is the reason for room temperature generally we use that lab 6 filament. Here I want to cover one important part, that is better we should know what it is, that is called so as I mentioned previously that okay, if there is a filament and filament it will merge here, this is our Winot cylinder. If Winot cylinder is there or Winot cylinder is not there, it doesn't matter. Even if there is a field emission gun there will be two anodes. Finally this electron beam it will come and merge somewhere here and then it will go like that. So if we have an optical axis, this will be our solid angle, that alpha I, and this will be our D0. Here current density J, it is depending on two part, one A T C square e to the power minus phi k t. It is a Boltzmann constant. A is a constant again, and this constant is depending on the metal, what metal we are using. So the current density, basically we are considering this is called the current density. So J, current density is denoted by J, and which is equivalent to A T square. So this Tc means temperature, what temperature we are heating the metal, and then that phi is, so this is work function again. Okay. So this A, this constant is depending on the metal, what kind of metal we are using, it will vary based on the metal. Then temperature, then heat, we already discussed about these things, that heat energy is very very important for our extract that electron, and then it is depending on our work function also. So this is the formula to get the current density. If we get the proper current density, that will help us to do our rest of the experiment with this current density. So whenever we will set up the instrument or install the instrument, there are couple of things we have to pay attention, that what will be the emission current, what will be the bias voltage, what will be our total current density, that during the installation time we have to pay attention. Otherwise most of the time we don't have to pay attention when we will do the imaging, but we have to know what kind of lens do you want, what kind of filament do you want to use it, that do you want to use it lab 6 filament, do you want to use field emission gun or XPEG, what is the difference between XPEG and cold PEG, what is the difference between the tungsten filament and lab 6 filament or Schottky filament. Better to know those things before we use those instruments for our biological science sample purposes. So here one more thing I want to add, that for the field emission gun most of the time we are using two anodes. So anode 1 and anode 2 which is helping to pull out the electron, and we are using a normal tungsten filament. We are giving the heat energy, but same time this two anode is helping us, so we do not have to increase very strong heat energy there to extract the electron, because there is a very strong electric field is there which is helping us to pull out the electron. So that is the reason, if you look at the work function for the field emission gun in that particular table what I was discussing at the beginning, if you look at the work function of this field emission gun, so it is written 4.5 and which is almost equivalent to our tungsten filament. Then how the field emission gun is helping us? Field emission gun is helping us because of this two anode which is helping us to pull out the electron properly. Okay, so now let's take a look about the different types of filament. It will really help us to understand what we are talking so far. So if we look at this filament, you can see that there are three major parts of a thermionic gun. So the thermionic gun we already discussed that here this is the filament cable. So this is the filament which is attached with the high tension cable. So high tension cable is attached with this filament and this is the filament tip, and there is a cap which will go and cover this particular area. This is called the window cap, and your filament it will come out from here, and at the top you have the anode plate like this. Now if we want to compare between the different types of gun, so then we can consider that the lab 6 crystal can be produced with a very fine tip about 1 micrometer in radius, which accounts for the smaller crossover size. Now we know what is crossover size. As a result, lab 6 current density are considerably higher than for tungsten, and the brightness is typically 10 times greater than the lab 6 filament. So even though lab 6 is usually operated at a much higher temperature to increase the life, so the decreased source size also result in improved the coherence and the energy spread. So these are the two important points, that how much energy spread is there, so how much it is spreaded and how coherent it is. So lab 6 is highly reactive and the gun vacuum has to be good enough to minimize oxidation during the operation. That is one of the main point. As I told that lanthanum hexaboride is a very good filament and it is 5 to 6 times expensive than normal tungsten filament. Now if your vacuum is not good, if there is a lot of oxygen or moisture is there, it is going to react with your lanthanum hexaboride and it will oxidize the crystal and crystal will break immediately. So for normal thermionic source wise, nowadays we are always using this lab 6 filament. Now if you compare to field emission gun, definitely field emission gun, as I mentioned that field emission gun or Schottky emission gun, your brightness is much higher, it is superior than other two filaments, and the beam will be much more coherent and it will help for high resolution TEM as well as our Cryo-EM also. If we look at that, okay, so there is a filament heating current. So this is the filament heating current, that we are heating a filament, and that emission current, that when we are heating a filament, is it going to immediately release the electron? Probably not. It is not going to release the electron immediately. So initially we need some kind of heat energy. If we just consider that, okay, so most of the time I am talking about that, okay, we have that tungsten filament. I told that for the tungsten filament, for tungsten filament we need this kind of heat energy. Okay, so here if we just even go 0 to 1500 Kelvin, you will not get enough electron, you will not get any electron. So you can see that if this is a filament heat current and this is the emission current, that filament heat current means how much heat energy you are giving, and then out of that what electron is coming out from that filament. So that is called the emission current. So if you just measure the emission current at the beginning, so you can consider from 0 to almost 2000 Kelvin, there is no emission. Then there will be a sudden emission. So you can see there is you can get an huge emission here, and then it will reach to the saturation point. Even if one day you can notice that, okay, your filament, you don't have enough brightness. So sometime what operator or users is doing, they are just constantly increasing heating of the filament. So even if you increase the heating of the filament, if you just go to 3500 Kelvin, it is not going to increase your brightness, it is not going to change your emission current anymore. And what will happen, it will reach near the melting point of this metal. The melting point of this metal is somewhere here, 3650 Kelvin, and it will melt the filament and it will break the filament. So what kind of heat energy is required to get the proper emission, that is depending on that particular filament, and also depend on the vacuum and microscope condition. Based on that, we have to play around with those parameters. So it is not a good idea, if your brightness is low, you just increase the heat energy of the filament and get the better brightness. It is not going to work in that way. So here I am showing a example of a lab 6 filament, where you can see that early stage, even if you switch on the microscope and try to see it at this particular temperature or this particular temperature, what kind of distribution you can see it. In this particular temperature, you can see this kind of distribution, where you have a center part, you can see a very dark point, and that is basically tip of that filament. It is an unsaturated electron beam, where you can see that tip of the filament and outside some kind of bright electron beam. So if you gradually heat and increase, so here heating should be a gradually heating, not in one go from 0 to 3000 Kelvin. So heating will be always gradually, it will increase, and that will help to heat the filament and get a proper emission current. When filament will be completely saturated, you can see this kind of electron beam in your microscope. Okay, that I already discussed when I was drawing that. Okay, when you have no bias, means if there is a no V-node cylinder here, there will be no bias, beam will be spreaded all over, some of the beam will be blocked by this anode and some of the electron it will go. You will not get enough, definitely maximum current it will come, but it will be diverged, it will not form a particular point here. So D0 is spreaded all over. So now this is the best scenario, where you have a optimum bias and intermediate current, and here if you have a high bias, most of the electron beam will block and then there will be no current. So here again you can see this is the emission current and the bias voltage. What is the relationship between emission current and the bias voltage? Biased voltage means V-node cylinder voltage. So what kind of cylinder voltage we can use it and get the maximum brightness. So if we don't have any bias voltage, means if there is no cylinder cylinder, someone can expect that most of the electron will come out. It is not going to block or convert somewhere. We can get the maximum current, but is it going to give the maximum brightness? Probably no. So even if you see that, okay, so this is the relationship between the brightness. So at beginning, emission current is starting from here, and that bias voltage, you will get the maximum brightness is somewhere here, where you have some kind of bias voltage here, and some kind of emission current, where you will get the maximum brightness. This is the curve it is showing you for the brightness. So where your emission current, it will decrease because you have the bias voltage, this one, your emission current, it will decrease, but your brightness is going to increase, and that is the reason we always try to follow this condition, will get a optimum brightness. Now measuring the beam diameter, that is also an important part. As I mentioned that previously, that when we have a filament, electron, it will come and merge in one particular point, and if you have a bias or not, it doesn't matter. If you just consider that, you will get a bright spot. So from this one, entire electron is coming and going through your rest of the column. So it may be the C1, then it may be the C2, and so on, and then it will give you an image, right? So in this particular case, if we just consider that measuring beam diameter, how we can measure the beam diameter? When that point source, this point source of electron, it will go through your C1 lens and C2 lens. C1 lens, as I mentioned, it will be condenser lens 1, and C2 lens will be condenser lens 2. So it will go through the condenser lens 1, condenser lens 2, and then it will give us an image on your sample, or if you don't have the sample, the same wave function can continue and give us an image on the screen. That is our first screen where we can measure the beam diameter. And how we can measure that beam diameter? So entire electron, it is coming, and based on this magnetic field of C1 or C2, we can get what is the diameter of this electron beam. So here we are just using the C1 lens, and different type of magnetic field we can use it for C1 lens, which is controlling the beam diameter. Here you can see, if we are using a very low magnetic field for C1 lens, you are getting a much wider beam diameter, where the beam diameter is much bigger. But if we are using slowly stronger magnetic field, where you can get a beam diameter, it will be shorter and shorter. So now rest of the lens is coming into the picture, which is helping us to increase the intensity as well as the beam diameter and so on. So gun lens is the first lens of the microscope, and then it is coming to the condenser lens 1, condenser lens 2, and condenser lens 3. So in this particular scenario, it will be better to know the lens system, how this lens system is helping us. So we are going to discuss about the lens system in next part. Here you can see, this is our filament. As I mentioned, this is our tungsten filament. This tungsten filament, this is the two part which is connected with our high tension. Okay, this is from the front view and this is from the side view, and you can see this is a very sharp hairpin region where electron will come out. So always that coherent electron beam, it is depending from where the electrons are coming out. So there will be two different types of coherency will be there. One is temporal coherence and another one is the partial coherence. It is depending on that what particular position electron is coming out as well as the energy of the electron. Is that different energy is also going to affect that coherency of that electron beam. So here that very sharp tip is very much important. If tip is not very sharp, if tip is broad like that, so electron can come out from this particular position or this particular position, and then there will be a difference, path length, the difference between this electron and this electron, which will not make the beam very coherent. So that is the reason if we just assume that all the electrons are coming out from the tip of the filament, we can assume that okay, the most of this electron beam will be a coherent electron beam. What about the lab 6 filament? So this is, you can see this is a filament of the lab 6 crystal. So this is a lab 6 crystal. So lab 6 crystal, it will look like this one, that very solid filament which we can mount, and here the high tension it will be connected and which will illuminate this lab 6 filament. There are a couple of images. Here I am showing you a filament that I already showed from the book, and this is a real picture where you can see that thermionic emission filament. This is the top part of that filament. So let me just so here you can see this is the tungsten filament which is connected with the high tension, and this is the filament tip which is mounted with this cylinder, where at the top part, here the top part is open, that's why we can see the filament, and here you can see the cover of the Winot cylinder and where filament is coming out from this tip, and this is called the Winot cylinder. This is the Winot cylinder where electron is helping out to pull out. And now this is the field emission gun. You can see that field emission gun, it is a very sharp tip and the electrons are coming out from the tip. We are always expecting that electrons are coming out from this tip, and the electron beams are mostly very coherent electron beam here. And this is a side by side comparison, so you can see that what kind of differences are there between the tungsten filament. So this is one of the tungsten filament where you can see this is both the cases we can use it for thermionic emission. One is that lab 6 filament and the tungsten filament. We have an anode which is helping us to extract that electron, and this is our Winot cylinder. So this tungsten filament and lab 6 filament is part of the thermionic emission. On the other hand, if we just consider that X-FEG and the normal cold FEG, both the cases there is a two anode are connected which is helping us to extract the electron from the tungsten filament tip. So this is all about the electron gun, and the next class we are going to discuss about electromagnetic lenses, how these electromagnetic lenses helping us to image. So today's class I'll stop it here. Thank you all for your attention.